All right, everybody, this is AP Bio Unit 5. We're going to start non-Mendelian genetics today. This will be Unit 5, Topics 4, 5, 6. I probably won't get all the way to the end of that in this video, but we'll go ahead and get started. Okay, we so far, I know we've dabbled in some of the homework assignments I've given you in some non-Mendelian, and we've kind of like flirted a little bit with some sex-linked traits and things like that. But let's get into covering those formally today. Okay. Now, those Mendelian ratios, really well-known Mendelian ratios, 100%, 50-50, 3-1, okay? Some traits do not follow those well-known Mendelian ratios that you should kind of have a pretty good idea of. Now, there are multiple reasons why, and you should have some familiarity with these, okay? The first one are degrees of dominance, okay? In everything we've looked at with Mendelian, those traits have complete dominance, okay? The seeds are green or yellow. They're not a mix. I don't even know what you mix green and yellow, but it doesn't sound pretty. The plants are short or tall. They are not medium, okay? The flowers are purple or white. They are not pink, okay? So some traits have varying degrees of dominance. The most commonly studied one in introductory biology is flower petal color. Okay, you cross a white and red flower, you get a pink flower. Animal hair does this as well. You cross a black and white mouse and you get like a brown mouse or something like that. Okay? The next one. Many traits are produced through multiple genes. Human hair and skin color are examples of this. And so are things like eye color as well. A lot of times you'll get biology students who will learn the Mendelian laws of genetics and go, Mr. Sater, they don't work. My eye color and my parents' eye colors are messed up. So don't try to use Mendelian genetics for non-Mendelian traits like the ones I'm talking about. Okay, some traits are on the sex chromosomes, so we don't really follow Mendelian genetics for those. We'll get into those very specifically. And finally, actually not finally, almost finally, some genes are so close to one another that they segregate as a unit. So some traits are inherited very closely together. And we'll look at some math on how to figure out how that works today. And finally, some traits are from what we call non-nuclear inheritance. And that just means the DNA for these traits are not in the nucleus. Well, where would they be? Remember, mitochondria have their own DNA. So the traits that your mitochondria have are inherited exclusively through the mom, because you always inherit mom's mitochondria. In mammals, actually I think it's almost anything that reproduces with sperm, including plants, so pollen. And if you're a plant, that also includes your chloroplast. All right. Everybody with me out there? Sorry I was a little bit late for the online session today. I'm going to move up. Maybe that'll help you guys a little bit. All right. Okay to go? Or wait? See a lot of pens still moving. I'm going to give it another second. People at home, you okay? Good. All right, I'm going to go ahead and jump forward. All right, we're going to break these down. So if you didn't get everything completely written down on that last slide, don't worry, because we're going to talk about them individually. Okay, we're going to talk about them individually. The first one we're going to talk about are degrees of dominance. Okay, this is easier than it sounds. Okay, in our Mendelian traits, things were either dominant or recessive, period. Okay? So he worked with traits that had complete dominance. Okay? These guys have the exact same traits, even though they are heterozygous and homozygous dominant. Okay? So let's say that white flowers are dominant. Big W would be the exact same thing as big W, little w. So they have the same exact phenotypes. In incomplete dominance, neither one is fully dominant, and the big key word is mix. Okay? You get a mix of traits, and flower colors are the big one. Okay? So you would see red plus white giving you pink. That's a mix, okay? It would not be red flowers that have like white dots or streaks on them. That would be something different, okay? Mix, mix, mix is the key, especially with colors. All right. The next one is codominance. Now listen, students mix these up. Okay? Just kind of want to go back for a second. Incomplete is a mix. Codominance is at the same time. So incomplete and co are both happening simultaneously, but in different ways. Okay? The best example for introductory biology students of codominance is two traits being expressed at the same time is blood type. Blood types in humans are A, B, and O. There are some other ultra rare ones, but these are the main ones. Okay? So you can have type A blood, type B blood, and the O I want you to think of as like a 0, okay? Type O blood is not really a type, it's a lack of, I mean, it is a type, but the O is like a lack of A and B, okay? So it is possible for humans to exhibit both at the same time. Okay? So type AB blood is not a mix, it is both. Okay? So the key word here is both. Okay? If we are co-captains of the football team, we are both captains. Okay? All right. So when you have codominance, You have to have genes that exist with more than 2 alleles. If you go back to the incomplete dominance, you're either white or pink. I'm sorry, you're either white or red, but both at the same time mixed together gives you pink. Okay? So they mix. With this one, they both exist simultaneously. So very commonly confused by students, which is why I'm kind of like being very explicit, slowing it down. So there's your examples up there. Okay? The alleles are, that's supposed to be a capital letter I, okay? And you can have the little A and B attached to it, or the recessive trait is I. So if a person is, if your parents are AB, there's different ways that can work out. Let's take a look here, okay? Let's do a quick practice problem. All right, you've got a black mouse, big B, with a white mouse, little b, and the result is gray. What is this? Is this co or incomplete? That is incomplete. You are right, okay? Incomplete because it's a mix, okay? All right, one more little practice problem in the middle here. Cattle can be red, white, or roan, which is some kind of mix here. Okay, what's this one? This is codominance because the colors have mixed. Okay? All right. Let's do it. All right, we've got a red cow crossed with a roan cow. What would the phenotypic ratio? One to one, red and roan, okay? Erase all this. All right. I always forget that I put them on there, and I can just... All right, one more. A woman with type A blood has a child with a man who has type B blood. That's very limited. You don't really know their actual genotypes. What could the woman be? What could the man be? All right, there's a couple possibilities here. The woman can either be big A, or just big A, and that right there is type O. I'm going to draw it in just so you see it. And the man's going to be the same way. Big B. There is no little b for blood type, okay? It just is its own allele. And so if these two people were like this, they could have children that are AB, A, B, or O, okay? O negative is the universal donor, but it can only receive type O. So it's like you're good to give, bad to get. Okay? When they come to the emergency room and they need blood right away, you give them O negative at first because anybody can take it. That's a whole separate set of genetics. It's just a different factor. It's called the RH factor. It's either you have it or you don't. And that's why it's either positive or negative. It's a protein on the surface of the, and there is more to blood type than those two things, but it's enough to donate. All right. Let's look at some things that are controlled by multiple genes. Good questions, everybody. So, the large majority of traits are controlled by more than one gene. Okay? A good example of this is something called epistasis. Okay? And what happens here is, these are called epistatic genes. The gene at one spot totally controls whether another gene turns off or on. Okay? It's almost like if I go down in the basement and I pull out, shut off the breaker, what does the light switch upstairs do now? Nothing, right? So the breaker in the basement has to be turned on in order for the light switch upstairs in the living room to work. Okay? That's how I think of epistatic genes. One gene has to turn on before the other one can turn on. Doesn't even matter what the switch upstairs is if the breaker's not on. Okay? So, here's an example. Coat color in labs. I don't even mean like science labs, I mean like the dogs. Okay? And some mice. Okay? One gene codes for pigment. Okay? One gene codes for pigment. And the second gene determines whether or not that's even going to be on. It doesn't matter what pigment they have if the epistatic gene is not activated. So take a look at these mice here. These mice that have little c, that's the epistatic gene. If little c is the trait, it doesn't matter what the other gene is, they're gonna be albino. Okay? So these albino mice, they have the gene for the hair colors, but the epistatic gene won't even let the hair color gene turn on. So this mouse right here, the breaker, okay, the fuse box, whatever you want to call it, is shut off for hair color. Okay? They have the gene, but it won't activate because of this other factor, this epistatic gene that will not turn it on. This is a kind of more interesting thing to look at with genetics because genes control other genes. Okay? Genes control other genes. All right. So you can get some weird ratios with these ones. All right, this one's kind of cool. This is polygenetic inheritance. Okay? If someone says, well, that trait's polygenic, that means more than one set of genes control it. Okay? This is what we're going to say about human height. Okay? And skin color. Okay? Your parents might be tall, but that doesn't mean you're going to be tall for sure. Okay? There's so many genes that control it, and some people who are very, very tall have more of those genes activated than people who might be shorter. Skin color is the same way. Okay? Take a look at this cross up here. It's got 3 alleles in it. People can be vastly different skin color than their parents. There's a really famous Instagram photo going around of twins where one parent has very, very dark skin, the other parent has very, very pale skin, and the twins, one of them has very dark skin, and one of them has very pale skin. So there's a lot of variation in human skin color, okay? It is controlled by multiple alleles. So skin tone can be all over the place and can be very different than parents. All right. You have to know that these are common polygenetic traits, but you do not have to know how to figure them out. Okay? So they might give you a question where they ask you about skin tone and why there's so much variation, and you would say one reason might be that it's a polygenetic trait. So that would be the limit of your, can you define it and know that it's an example? All right. I'm pretty sure my PowerPoint color scheme does not represent human skin tone, especially number 4, like orange. All right. The next one, which you've probably dealt with before in your introductory biology class in high school, is sex chromosomes. Okay, there's a couple cool things going on here. First of all, back when we looked at karyotypes, how big is a Y chromosome compared to its partner X? It's very small, about half the size, and it doesn't have 4 branches like an X does. It only has 3. So, sex-linked genes are genes that are found on that 23rd pair. So, this guy named Morgan experimented with fruit flies who only have 2 chromosomes, which made it pretty easy to figure out. If you have 23, this was before DNA analysis and gene mapping, he determined that traits can be carried on sex chromosomes. They don't just, I've kind of told you this already, they don't just control like your sexual characteristics. Women don't have a prostate gland, but you carry the genes for one, okay? So not everything about the sex chromosomes is on the 23rd pair. Just make sure you understand that. So, sex-linked genes are either on the X or Y chromosomes. There are very, very few Y-linked genes, okay? There are some genetic disorders where you can survive with one X chromosome in the sex chromosome set, but you cannot survive with only a Y chromosome, okay? Because there are missing genes that are on the X-linked. So there are very few Y-linked genes, so we don't see a lot of Y-linked disorders. Okay. Everybody has at least one X chromosome in the sex chromosome set. If you're female, you have 2. So because everyone has at least one, those ones are easier to track, and there are several genetic problems linked to sex chromosomes in humans. So a couple things are going to be going on here. Fathers can pass X-linked alleles to all of their daughters. Okay? Let's do this real quick. If you're XY and you're female, which one of those came from your dad? Only the X. It's not like you get both of your moms. You get one from your mom. Here's mom. You're going to get one of these over here on the side, okay? And you're going to get one of these. So if you're dad here and you have X-linked traits, they go to the girls. Okay? If your dad, if you have sons, the only place a son can get an X chromosome from is from mom. Okay? Dad gave the Y, so the only place to get it from is mom. And that's what the next point is. Moms can give their X-linked genes to both types of children. Okay, let's take a look at this. This is where we draw the X's and Y's and we put little subscripts or superscripts on. Okay? So there's some recessive trait that's affected in the father. You see that little N right there? Okay? All of his daughters are going to be what? Homo or heterozygous? Yes. There they are right there. All right, so all the females are carriers now. What are the what if we take those gals right there, their sons might have trouble. Because their sons have a 50-50 chance of inheriting the recessive gene. All right, so you just got to keep in mind that, like, well, the Ys don't have a copy, so they are going to get what they get. Okay, they get what they get. All right, a little bit more about X-links, okay? Females only express the trait if they are homozygous. Females that have heterozygous recessive are very rare, okay? Very rare to have female colorblind, okay? It can happen, but it's very rare. Because both mom and dad would have to be colorblind and mom would have to be a carrier. So males, we only have one copy of those genes, so if there's a much better chance of us having it than females. If you only get one copy, you get what you get. If you're female, you might have a good one. Okay. Here's a couple X-linked disorders. Duchenne's muscular dystrophy. Okay, your muscles get weaker and weaker and weaker as you go through life. It's a protein on the X chromosome. Often found in royal families, especially of the czars of Russia. Hemophilia. There's many different types of hemophilia, but there is one that's X-linked. Color blindness, we already mentioned. Did you guys get those? Those are the common ones you'll see. In our class. They may come up with one I've never heard of. It doesn't really matter if you've never heard of it, okay? If it has a big, long, complex name, don't let that scare you. All right, let's look at some other interesting things about X-linked chromosomes, sex-linked chromosomes. Okay, in women, we have to do something to one of their X chromosomes, okay? Women inherit 2 X chromosomes, so you have double the amount of X that males do. So during female development, most of the X chromosomes in each cell become inactive, okay? And it becomes this little thing called a Barr body. It condenses, and this helps regulate what we call gene dosage, okay? We don't want that gene to be overactive, so one of them will condense down and become an inactive chromosome. Those are called Barr bodies. Hey, would you mind pulling that door shut? Gene dosage is a concept we don't really get into in this class, but we don't want to have that gene be overactive, okay, because there's 2 copies of it. All right, linked genes. This gets a little complex, okay? Linked genes, we're going to start getting into some math. Let's take a look here. Okay, this is a review. Genetic recombination is where we get offspring with new combinations of genes from parents. In other words, you have a copy from mom and a copy from dad, but those copies are scrambled due to multiple reasons that we have already learned, okay? So if you have the exact copies of your parents, we're going to call you a parental phenotype. Recombinations are going to be where genes have phenotypes that are different than the parents, okay? And this is a good thing. It gives us variation, but not all genes segregate according to Mendelian rules because they're close to one another. Was that too fast? I'm sorry, I didn't hear you. They are on Schoology, and I'm going to put this video up a little bit later. And if you're watching this video right now, you can hit the right button to skip what this little pause for my students. Just be patient with us. All right. You guys feeling comfortable? There we go. All right. So Mendel observed some recombinants during his crosses, okay? When he did a green wrinkled cross with a yellow round, he found that they could combine in ways where green and wrinkled didn't follow each other. There was a combination that was new, okay? So he took this recessive green wrinkled and crossed it with this heterozygous for yellow, heterozygous for round, and when that happened, what we get are 50% of them match the parents. Okay, do you see how they match the parents? Well, if you don't see that, this matches and this matches. Now we have a recombination, a new combination. Okay, we have yellow that's wrinkled and we have green that's round. So these traits did not travel together. These are recombinants. Now, this is a Mendelian ratio, 50-50. If they are 50-50, that tells us that they are unlinked or that they are on totally different chromosomes. Unlinked means the genes might be on the same chromosome, but they're so far apart that they still sort out randomly. What do you think would happen to the number 50-50 if the genes were very close together? Well, they always have to add up to 100%, but what you're going to see is they're going to become non-random. They're going to become closer together, and we call the units that those separate by map units. Map units are not a physical distance, they are just a percentage of change. And so before we could map out genes with modern science, people started looking at the mathematics of recombinants and noticed that some of them were not 50-50, and some of them, the traits matched up in different frequencies. The closer those were together, the closer the two traits were to being not 50-50, the closer the genes were. Okay, let's take a look. We call these linked genes. Linked genes. All right. Linked genes are located near each other on the same chromosomes, and they tend to be inherited together. And the closer they are together, the more likely they are to be inherited together. So, a lot of things create randomness, okay? Independent assortment of chromosomes. If things are on different chromosomes, they cannot have any linkage whatsoever, okay? We have crossover, and we have random any sperm, any egg, okay? So we have all this randomness. But if we take out small amounts of the randomness, because they're on the same chromosome, and because they're close together on the same chromosome, we get linked genes. Okay. Here's an example of some crossover. That would mean there would be no recombination, non-recombinants. Okay. If you look here, we've got some C's, okay? We've got big C here on the blue and little c here on the red. They cross over, and each one gets a big little and a big little. Those became heterozygous. Those genes are far apart. They're far from the centromere, okay? So these ones cross over, but they're going to be of course, these change the same segment, okay? These change the same segment. Let's look at crossover a little bit deeper here. The farther apart two genes are on the same chromosome, the higher the probability that there will be a crossover, and the higher the probability there will be a recombination frequency of 50-50, okay? So, was that too fast? Tor, are you good? Okay. So crossover explains why some linked genes become separated and they're not completely linked. So the big sort of like hypothesis behind this, the big sort of theory behind this is that when genes are far apart, they have a higher probability of crossing over. When genes are close, they will stay together. Okay, so there were some experiments produced by a guy named Sturtevant, and he allowed scientists to map genes without us actually knowing where they truly were on the chromosome like we do today. Right now we know exactly where it is. We can go and, find that exact spot. At this time we didn't know it, and we deducted it by indirect measurement using math. Okay? So this gets a little complex here. We started making something called linkage maps. A linkage map is based on recombination frequencies, okay? And the distance between genes was measured in this unit they came up called a map unit. And it sounds very complex, but it's equivalent to 1% recombination frequency, okay? So that would be very low. Those would be linked almost 100, exactly. They don't recombine at all. This gave relative distance, okay? I can arrange all of you by height and not know how tall you are, okay? I need to know exactly how tall you are. I just need to know, who's the next tallest. That's relative distance. So we really didn't know the true distance. We just know that this one's farther apart than this one. Okay, so all we could really say with this technique was these genes are so far apart that they never recombine. That would be 50-50. Or these genes are so close that they always recombine. So what percent of the time do they create a new combination? The max is 50-50. The lowest would be 0. Fully linked or completely non-linked. Now remember, genes on different chromosomes never have linkages, because during meiosis we have random assortment, okay? So genes on different chromosomes never link. When you start really studying large populations of traits, you can figure this out, okay? You can figure this out. You're not going to figure this out with a classroom full of people. You have to examine a lot of people. Okay. Oops, sorry. These numbers represent how far these traits and fruit flies are from these things called aristae. And so short and long are the same gene, and the farther you get away from it, the more likely it is to have recombination. Okay, I'm going to stop here. I have a sheet on Schoology that walks you through map distance very slowly. It's on Schoology. In today's folder, it's HW genetic mapping. That's what I'd like you to work on for the rest of this period. I'll help you with that.